Welcome back to the Deep Dive. Today we're digging into some fascinating research, really getting under the hood of a major scientific paper. We're tackling one called Airborne DNA Reveals Predictable Spatial and Seasonal Dynamics of Fungi. Big title, big team too. Lead author Nerea Abrego. But it's a huge international effort.
It really is. And this paper, it's not just about listing fungi. It's trying to settle a pretty old debate in science.
Which debate is that?
Well, whether microscopic life, fungi, bacteria, all that invisible stuff follows the same big global patterns, the same rules as like plants and animals, macroorganisms.
Ah, right. Like the classic pattern where biodiversity, the number of species, increases as you get closer to the equator, the latitudinal diversity gradient.
Exactly. But for microbes, the thinking, or at least some of it, was maybe they're just everywhere. Tiny spores blown all over the planet. So do fungi actually follow those big biodiversity rules?
And finding out matters, doesn't it? Fungi are, well, they're everywhere and do everything. Decomposers, partners for plants, sometimes pathogens.
Absolutely crucial. But we know surprisingly little about where they actually are globally. The estimates for how many species exist are just wild. 500,000, 10 million.
Wow.
Yeah, and we've only formally described about 156,000, so we're working with maybe, less than 2% of the actual diversity out there. It's kind of a black box.
Okay, so before this big study, what was the picture like? You mentioned soil studies.
Yeah, mostly soil studies, and honestly, they were a bit of a mess. Some found more diversity near the poles, some near the equator. The results were all over the place, really conflicting.
So a clear knowledge gap then.
A big one. And that's where this study comes in. The real innovation wasn't just the DNA sequencing, though that's key, but the method. They used standardized global air sampling.
Air sampling, so collecting spores and stuff out of the air itself.
Exactly. Spores, bits of hyphae, other airborne fungal bits. The idea is that air sampling gives you a more integrated picture, maybe captures the dispersal phase better, shows stronger ecological signals than just digging in the dirt in one spot.
Okay, let's talk about how they actually did that. This Global Spore Sampling Project, or GSSP. How massive are we talking?
We're talking properly global. 47 sampling locations spread across every continent except Antarctica, really trying to cover the different climate zones.
47 sites. Wow. Tropical, temperate, polar. How did they keep the sampling consistent everywhere? That must have been a challenge.
Standardization was absolutely critical. They use these things called cyclone samplers. Basically, they suck in a huge amount of air and catch all the particles on a filter.
Right.
And at every single one of those 47 sites, they collected samples for 24 hours, every single week, for a year or even more.
Every week, for a year.
Or more, yeah. That level of consistent long-term data, that temporal replication, that's one of the real strengths here.
Okay, so mountains of samples. Then the lab work. You mentioned DNA. They targeted the ITS region. Why ITS specifically? For listeners who know DNA but maybe not fungi specifics.
Right, ITS. It stands for Internal Transcribed Spacer. It's a little bit of fungal DNA, part of the ribosomal genes, that changes pretty quickly between species. So it's variable enough to tell species apart, but the areas around it are stable enough that you can design universal primer starting points for copying the DNA that work for almost all fungi. It's like the standard barcode for fungi.
Got it, the universal barcode. And processing all that data gave them just over 27,000 OTUs. Explain OTUs quickly.
Yeah, operational taxonomic units. Think of it as a practical way to count species when you're using DNA sequences. You basically group sequences that are super similar, usually like 97% identical or more, and call that group one OTU.
So a working definition of a species based on the DNA sequence.
Pretty much. It's a consistent way to measure diversity across lots of different samples, even if we don't have a formal name for every single sequence type.
Okay, so 27,954 fungal species, plus climate data, plus data on the fungi themselves, like their ecological role, spore size. They really layered the information.
Exactly. That integration is key. It lets you ask why things are where they are, not just what is there.
All right, let's get into the really juicy stuff, the findings. This global map they built. What did it show? Were fungi just everywhere like some people thought?
Nope. Not at all. The data showed really strong geographic limits. Most of those OTUs, the vast majority, were only found in one climate zone. Polar, temperate, or tropical.
Really? Only one?
Yeah. Only about 3.5% were found in all three zones. So that idea of microbes are everywhere, It really doesn't hold up for most fungi based on this air sampling. They're much more restricted.
Okay, so if it's not just about being blown everywhere, what's keeping them in check? What's the main environmental factor controlling where they live?
Temperature, specifically mean annual air temperature, or MAT. This was probably the biggest single finding.
How clear was that signal?
Incredibly clear. They looked at precipitation, MAP, aridity, and MAT alone explained 78% of the variation in fungal community composition across all those sites.
78%, just from temperature.
Yes.
That's massive for an ecological study.
It's huge. I mean, precipitation mattered too, explaining 42%, and aridity 35%. You obviously need water. But temperature seems to be the fundamental filter, setting the broad geographic boundaries.
Wow. Okay. And does that temperature control link back to that big biodiversity pattern we talked about earlier, the latitudinal gradient?
It does, perfectly. The study confirmed fungi follow it. Overall diversity measured by both the amount of fungal DNA in the air and the number of OTUs. The species richness, it clearly increased as you move towards the equator.
So more fungal species in the tropics.
Just like
plants and animals.
Exactly. Tropical areas had the highest diversity overall. It fits the classic pattern beautifully.
But were there any exceptions, any groups that bucked the trend?
Yes, and they're interesting ones. Fungi that form lichens, certain types of mycorrhizal fungi that partner with specific plants like heathers, ericoid mycorrhizae, and also the big group of ectomycorrhizal fungi.
The ones that associate with many forest trees.
Those ones, yeah. All of those groups actually peaked in diversity in the temperate zones, not the tropics, which kind of matches what some soil studies have hinted at before.
Interesting. So different functional groups have different hot spots. But that raises a point about the method. How well does catching stuff from the air represent all fungi? What about those ectomycorrhizal ones, ECM? Do they even release spores high into the air like pathogens might?
That's a really important question and a limitation we should definitely circle back to. Air sampling is brilliant for capturing fungi that rely on wind dispersal. And sure enough, the study found lots of plant pathogens, general decomposers, wood rot fungi, groups that need to spread far and wide on the breeze.
Okay, so it captures the wind dispersed specialist really well.
Right. Now let's shift gears a bit. From space to time, that weekly sampling for over a year, it gives this amazing window into seasonal changes globally.
And the pattern there was pretty stark too, wasn't it? Seasonality gets stronger the further you are from the equator.
Absolutely. The variation through the year in both the sheer amount of fungal DNA and the number of species was way more dramatic in the polar and continental zones compared to the tropics.
Can you paint a picture of that difference? Like polar versus tropical air?
Sure. Think of a tropical site. Fairly high levels of fungal DNA pretty much all year round. It's always warm enough, usually moist enough. Now contrast that with a polar site. For much of the year, the air is relatively quiet in terms of fungal DNA. Then, boom, during the short summer growing season, there's this massive, sharp peak, a burst of activity.
It's biological fireworks, just for a short window.
Exactly. A really compressed reproductive period driven by that steady arrival of warmth.
And did the types of fungi change more drastically season to season in those colder places too?
Yes, the community turnover, the rate at which species replace each other, was much higher from spring to autumn in those colder polar continental sites compared to the tropics over a similar period. The whole community composition fluctuated more strongly where seasonality was intense.
Makes sense. And zooming in even closer, did daily weather matter? Like a warm, windy day versus a cool, still one?
It did. They found that both the amount of DNA and the number of species detected were generally higher on warmer and windier sampling days. So you have the big MAP pattern setting the stage, and then the daily weather acting as the trigger for spore release.
Okay. Now, there was a really fascinating detail about spore size differences.
Yeah.
Something about sexual versus asexual spores.
Yes, that was quite cool. They found opposing patterns. Asexual spores, often for rapid spread, were generally largest in the warm tropical and subtropical zones.
Okay.
But sexual spores, often for survival genetic mixing, were largest in the colder temperate and polar zones.
Huh. Why would that be? Bigger asexual spores in the tropics, bigger sexual spores in the cold. Seems counterintuitive, maybe?
Well, think about the pressures. In the tropics, maybe a larger asexual spore gives you a head start, helps you colonize quickly in a super competitive environment. But in the polar regions, that short growing season, the long, harsh winter, a large sexual spore, even if you make fewer of them, is like packing a bigger lunchbox. It has more resources stored inside to survive that long winter and be ready to go the instant conditions allow. It's an investment in surviving the tough times.
That makes a lot of sense. An evolutionary trade-off shaped by climate. Okay, let's pull back again. How do these patterns, temperature control, seasonality, relate to the deeper evolutionary history of fungi? You mentioned they used some advanced modeling, HMSC.
Right, hierarchical modeling of species communities. It's a way to look at how species are distributed based on the environment and their evolutionary relationships, their phylogeny at the same time.
So it tells you if related species tend to live in similar places or respond similarly to seasons.
Exactly, and they measured this using something called Pagel's lambda, symbolized as U.
Lambda. Okay, what does that number mean? They reported a strong signal for seasonal sensitivity, Killy 0.39. Is 0.39 big?
It is, yeah. Pagel's lambda runs from 0 to 1. 0 means no phylogenetic signal. A trait is random across the evolutionary tree. 1 means the trait perfectly matches the tree structure. Close relatives always share the trait. So 0.39 is a pretty strong signal, meaning that sensitivity to seasons seems to be a trait that's conserved in some fungal lineages. It's sticky evolutionarily. Groups like, say, the Polypiralis bracket fungi seem to have inherited this tendency for a very pronounced, short sporulation season linked to climate cues. It's an ancestral adaptation.
So they're kind of hardwired for seasonality.
To some extent, yes. They also found a moderate signal equals 0.28 for overall chromatic sensitivity, which suggests that while ancestry plays a role in where fungi live, there's also been ongoing adaptation to different climates across the fungal kingdom. It's not completely fixed.
And interestingly, no signal for the exact optimal temperature or the timing of the peak season.
Right. That shows there's still a lot of variation even within related groups. Adaptation is happening. But that core seasonal response in some groups seems quite deep-rooted.
So, boiling it all down, the really big takeaway here is that fungi, this incredibly diverse, vital, but often hidden kingdom, they do follow predictable global rules, especially that latitudinal diversity gradient driven by temperature, just like the big stuff we can see.
Exactly. It really strengthens the argument that the fundamental rules of biodiversity apply across scales, from elephants down to microscopic fungi. It gives us that global blueprint we were missing.
But we need to touch on that limitation again. Those ectomycorrhizal fungi, the ECMs, if they're underrepresented because they don't disperse as much high in the air, how complete is this blueprint? Aren't we missing a really important part of the forest ecosystem, for example?
It's a fair point, and a key limitation the authors rightly acknowledge. This study gives an amazing picture of the aerially dispersed fungal community. ECM fungi might rely more on, say, animals eating truffles and spreading spores that way.
Like squirrels planting trees, but with fungi.
Sort of, yeah. Or maybe their spores are just larger and fewer, an evolutionary choice that means they don't get sampled as effectively by high-volume air samplers. So yes, the map isn't perfectly complete for all functional groups.
But the main conclusion about temperature still holds.
I think so, yes. Across the huge diversity they did sample effectively, MAT was overwhelmingly the dominant driver of spatial patterns. Airborne DNA gives this synthetic view, and temperature is the main theme coming through.
Which inevitably brings us to climate change. If MAT dictates 78% of where fungi live globally, and we're messing with MAT everywhere,
the implications are massive. Fungal communities are almost certainly going to restructure, potentially quite dramatically. This study gives us a vital baseline, a way to start predicting how those shifts might happen. It's crucial for understanding future ecosystem function.
Understanding the invisible majority, basically.
Pretty much all captured from sampling the air we breathe.
Amazing stuff.
It really is. And it leaves us, and you listening, with a really interesting question to think about. We saw that strong evolutionary stickiness for seasonality in some groups, like those Polypiralis. They seem adapted to a short, sharp season.
Right.
So as climate change shifts temperature zones and potentially lengthens growing seasons in many areas, how fast can those fungi adapt? Can a group that's seemingly hardwired for a short burst switch gears quickly enough to track those changing conditions, or will they struggle?
A big question about evolutionary lag in a rapidly changing world. Definitely something to mull over. We'll leave it there for this deep dive.